Welcome to the lecture, Synthesis of Triacylglycerols and Major Membrane Lipids. This is really the second part of the fatty acid synthesis lecture we just did. When you read lecture 38, all of this content is in that same chapter. So you're kind of getting two lectures out of one chapter here. And again, the lecture objectives are going to focus more on the triglycerides and membrane lipids and that kind of stuff. It will be helpful for you. All right, let's pick up with where we left off at the end of the fatty acid synthesis lecture. And there we knew we created some free fats. And the liver says, I don't want to store free fats. I'm not a fat storage organ. And what it wants to do is take those fats and create a triglyceride molecule and then puts the triglyceride inside the very low density lipoprotein particle, and it sends that out in the blood to be used or stored elsewhere. Well, how do we do that? Well, we create this molecule called phosphatidic acid. And phosphatidic acid is a very powerful molecule because, as you see, it's a glycerol, 1, 2, 3 carbons, and you attach two fats to it, and there is a phosphate group there. If you want to make a triglyceride, you knock the phosphate off and you put another fatty acid there, and you have 3 fats on the glycerol, or a triglyceride. If you want to make a glycerol phospholipid, you've already got the beginning structure now. You've got the glycerol backbone, you've got the 2 fatty acids as part of that glycerophospholipid, and then all you need to do is add a head group onto that phosphate group, and you've created the molecule. And glycerophospholipids will be used for VLDL particles because glycerophospholipids make that lipid shell of the particle, and we'll see that in just a few slides here. But phosphatidic acid could also be used to make lipids for the membranes and the plasma membrane or the organelles or whatever. So you see a variety of uses there. Well, where does that phosphatidic acid come from? Well, obviously the fatty acids are synthesized by the liver, but where does the glycerol come from? The glycerol backbone is going to be glycerol 3-phosphate, which is produced by either phosphorylating glycerol, which takes place in the liver. The liver has glycerol kinase, and it can phosphorylate it and give that molecule which is going to be needed to make this phosphatidic acid. Or in the liver and the adipose tissue, under an insulin signal, when blood glucose is higher than normal, there's plenty of glucose available, both liver and adipose will be in a glucose utilizing mode. They'll be doing glycolysis. You may remember glucose produces fructose 1,6-bisphosphate. And that 6-carbon molecule is split into 2 3-carbon molecules. There's a DHAP and a glyceraldehyde 3-phosphate. And the DHAP now can be used in a variety of ways in the cell. And one of the things it can do is it can be reduced. It can be reduced to glycerol 3-phosphate. How do you know it's reduced? Because you see NADH here. NADH is the reduced cofactor, and it's going to give its hydride to DHAP, which will reduce it to glycerol 3-phosphate, and in the process the NADH will become oxidized, and that gives you the oxidized NAD. So liver and adipose can produce the glycerol 3-phosphate needed to create the triglycerides. Now for liver, you're going to do that because you want to create the triglycerides that you put in the VLDL particle to export. In adipose tissue, you're going to take free fats and you're going to build triglyceride, but adipose is a storage organ for, or cell for fat. And so you create that triglyceride primarily to store it. Okay, so once you make those triglycerides, it's time to transport them. The liver says, I don't want to store triglyceride. I want to get rid of it. I need to send it out. And you send it out through these VLDL particles. So here's phosphatidic acid. You knock the phosphate off. You put a 3rd fatty acid on. 1, 2, 3 fats is a triacylglycerol. If you're a liver, you put that into a VLDL particle and you release it into the blood. If you're an adipose tissue, you make that triglyceride and you simply store it with the other fat portion in the cell, the fat droplets where it's stored. And we'll see the process for making VLDLs and how they're exported in the coming slides and coming lectures actually as well here. Now what's interesting about a VLDL particle, and we'll see this in a lot more detail coming up, you look at the percentage of the total weight of the particle. Triglyceride by far is the most dominant species. It gives most of the mass to this particle because that's its job. Its job is to transport triglycerides in the blood in a way that remains soluble and can get to the destination. There's a little bit of phospholipids because the phospholipids make a shell, and the polar part of the phospholipid faces the water medium, and the inside is going to be very hydrophobic. There's going to be a couple of proteins on the outside that help give the VLDL its functional properties, and of course triglyceride on the inside. Okay, so if you have a phosphatidic acid, you can make a triglyceride, and that happens all the time. But you can also make glycerophospholipids, starting with the phosphatidic acid. And it comes down to putting the head group onto that phosphatidic acid. And there's two major strategies for doing that, two major pathways. And it's kind of a lot of detail, probably a little bit of gobbledygook here. So I just am interested in understanding that there are two pathways. So the ways to do this are the phosphatidic acid has a phosphate on it. And if there's a phosphate on that third carbon and you want to put a head group on, there are two ways to do it. You can phosphorylate the head group and make it chemically active so that it's able to attach directly to the diacylglycerol once that phosphate comes off. Or it's possible to activate the phosphatidic acid using a cytidine triphosphate, a CTP. And it's the same thing we've seen a million times before. You knock a pyrophosphate off the CTP and you attach the diacylglycerol along with its phosphate onto that cytidine monophosphate to make a CDP. It's like UDP glucose. It's like the GTP that we saw before being used in the same context there. And then the head group comes in, it reacts with the activated diacylglycerol, and the head group gets transferred on to the glycerophospholipid. And so there are certain head groups that will operate through the phosphorylated head group, and there are others that will work through an activated phosphatidic acid. And I'm not interested in you memorizing that, but just understanding that there are two pathways here. And once you make these head groups, it's really interesting how you can interconvert these. In fact, if you see the interconversion here, these are the ones primarily on the left where you activate the head group and put it onto the diacylglycerol. And we'll see this in the amino acid section coming up pretty soon, that if you have a diacylglycerol and you add a head group, ethanolamine or choline, you can easily interconvert these. The ethanolamine head group has a NH3 on the end, whereas phosphatidylcholine has 3 methyl groups on that terminal nitrogen there. And so we'll see later that S-adenosylmethionine is a methyl carrier. We saw S-adenosylmethionine in the vitamins lecture. And 1, 2, 3 methyl groups go into ethanolamine to transfer this thing into a choline molecule there. And then we can also transfer ethanolamine into serine here by taking advantage of that nitrogen and putting a carboxyl group on there and changing ethanolamine into a serine, which is basically an amino acid that's attached through its chain to the phosphate. Again, more detail than what you need to memorize, but what I want you to learn is that just from starting with any number of starting materials, you can interconvert these different species and you can populate what's needed for a biological membrane. Some of these are a little bit more involved. These are the ones where you would activate the diacylglycerol first and then add the head group. And one of the ones we've talked an awful lot about is phosphatidylinositol. That's the one that has this carbohydrate attached to the phosphate. And this is the one that can be phosphorylated on this ring to make the molecule phosphatidylinositol bisphosphate, or PIP2. And you notice that it's still attached to the phosphate here. And that is the one that when it's cleaved by phospholipase C cuts the bond between the glycerol backbone and the phosphate. That will release the IP3 molecule that's important for signaling. Another example over here is the molecule cardiolipin. Cardiolipin is a really interesting molecule because it's almost like 2 lipids in one. You've got 1, 2, 3 carbons over here, you've got 1, 2, 3 carbons over there, and so it almost like forms 2 sets of lipids that are connected. But there's some structural changes there, but it's a really interesting molecule that has interesting function that we won't talk about at this level. Okay, remember the class of sphingolipids when we talked about those? Sphingolipids are based on the 18-carbon amino alcohol sphingosine. These are not glycerol-based, but they are sphingosine based here. And sphingosine is that 18-carbon, it's an amino group and an alcohol, amino alcohol. And you can add a fatty acid onto this nitrogen here through a amidation reaction. And when you add the fatty acid, you create one long hydrophobic tail here on the nitrogen. There's already a hydrophobic tail from the sphingosine itself, and that gives those two long hydrocarbon chains that are attached to a polar group up here, which is kind of like the head group on a phosphoglyceride molecule here. And then we know that we can then modify this. We can oxidize, we can put double bonds in this, or we can elongate, whatever we need to, just like we saw before in the fatty acid synthesis lecture. Where does the sphingosine come from? We can make that. Here's palmitate, a C16 fatty acid, and we have serine donating some of the atoms to the molecule, and serine, so serine and palmitoyl-CoA comes together to make that sphingosine molecule. Sorry, I got a little bit tongue-tied there. And to remind you what you can do with the sphingolipids, if you start with the sphingosine, that 18-carbon amino alcohol, and you attach a fatty acid through an amide linkage here on that amine group, you create a ceramide. This R group is going to be one of these long fatty acid-like molecules. So you see the two chains like a glycerophospholipid, but it's a little bit different. It can be shaped like glycerophospholipid, but that's not glycerol right there. And then we know that we can add a variety of head groups on. We can add a phosphate and a head group, and we can create the molecule sphingomyelin. And sphingomyelin is going to be very important for nerve cells and for the myelin sheath. And the way this thing is configured provides a certain amount of spacing that when these membranes wrap around each other, there's a certain amount of space between them that helps dampen any electrical current at all. And that's what myelin sheaths do, is they really protect against current loss during action potentials. And you'll see that in the neuroscience class. We know it's also possible to add carbohydrates on here, and if you put one carbohydrate on through a O-glycosidic bond, you create a cerebroside, but you can also add a polysaccharide on through an O-glycolytic bond here, and you create a ganglioside. So I remember this by saying it's really smart to add a sugar to a sphingolipid, so that's a smart cerebroside. Ganglioside is a gang of sugars. And based on what the structure of these sugars are, really does change the shape of what this lipid looks like when it sticks up above the membrane here. There's a really nice space-filling model. So it's not enough that these things are sitting in the membrane and just being a part of that bilayer structure, but all that interesting carbohydrate group that sticks up above the membrane provides sites of recognition where molecules that come in contact with the outer surface of the membrane will recognize these shapes and understand what part of the membrane they're in and whether they should bind or whether they should attack or modify or whatever. Really important for the structure and recognition function. So this is just all on one slide here about how you can make ceramides and gangliosides and different kinds of things. There's a sulfatide with sulfate groups there. Here's a galactose cerebroside where you're adding galactose instead of, say, a glucose molecule or anything else. There's a lot of different varieties here, and all of these things are going to have very specific functions based on what the cell is and what it's trying to do. And one of the things it tries to do is exactly this recognition here. We have glycoproteins. If you recall, those are proteins that have carbohydrate groups. We had the O-linked and the N-linked glycoproteins, and they provide a lot of recognition in terms of structure and function. And then if you see, a lot of times in between these glycoproteins, you'll have lipids that are also glycosylated. And those lipids with all of their structure will come together with the proteins to make very specific binding sites or recognition sites. And using fluorescence imaging techniques and some of the other ways to uniquely identify proteins and lipids, it's able, this is a cartoon obviously, but you're able to see now that there are these little pockets and these little rafts or these little areas that are composed of these glycosylated proteins and lipids that make docking sites where cells come together or proteins will bind to cells or some kind of cell-cell contact, or you have immunological cells that will tumble along the membrane surface here and it will recognize these patches, and it will recognize that's a patch where it can bind and sit and do some of the work that it needs to do. So that really important function here for the sphingolipids and the gangliosides and cerebrosides. Okay, so let's go back now to the liver, to fatty acid synthesis and triglyceride synthesis, and let's look a little bit more at VLDLs here. The big picture we've seen before, we know that when glucose in the blood is higher than normal and insulin is out in the blood, Insulin is going to tell the liver to take that glucose in. It's going to tell the liver to operate with a glucose metabolism, and the liver will use glycolysis to get its major energy. And pyruvate is the end of glycolysis, and since the liver has mitochondria, the pyruvate will enter into the mitochondria, enter TCA, electron transport chain, and oxidative phosphorylation. And because there's a lot of glucose, there's a lot of energy, and the liver is pretty happy. And so the liver says, well, what am I going to do with all this excess glucose? And even though it's not on this picture here, the priority of the liver is to make sure that its glycogen stores are filled up with the highest priority. Because as soon as the glucose goes away and insulin comes down, the liver has to turn over to a glucose producing mechanism. But assuming that the liver has done that, assuming the liver has synthesized all the glycogen it can hold, and there's still excess glucose that needs to be handled, the liver will have fatty acid synthesis going on. You're seeing all the processes here that you didn't see before. Under those conditions, there's plenty of energy in the liver. You don't really need a lot of TCA cycle activity because the ATP needs are met. And so when the pyruvate goes into the mitochondria, it can be converted into OAA and into acetyl-CoA to make both the molecules necessary to make citrate. Even if the TCA cycle is not running at a very high level, you can still produce that citrate. Citrate has the ability to leave the mitochondria because it has a transport protein. Citrate gets out into the cytoplasm, and it's split by citrate lyase into oxaloacetate and acetyl-CoA. Acetyl-CoA is going to be the starting material for fatty acid synthesis like we saw last lecture, and in a lecture coming up we're going to see it's the starting material for cholesterol synthesis as well. The OAA gets recycled to malate, malate to pyruvate. The interesting thing here is that you've got this malic enzyme which will do the conversion of malate to pyruvate, and in the process will produce NADPH. NADH is going to be a source of electrons needed for these biosynthetic reactions, both cholesterol synthesis and fatty acid synthesis. We know that NADPH is made primarily by the pentose phosphate pathway. We know glucose 6-phosphate dehydrogenase is the first enzyme of the pentose phosphate pathway, and it's the enzyme that will pull glucose 6-phosphate into the oxidative phase of the pentose phosphate pathway that leads to the production of large amounts of NADPH. But still, these will combine. The liver doesn't care where the NADPH comes from, it just cares that it has NADPH that can be used in the reduction reactions necessary to synthesize a fat. And what pops off of the fatty acid synthase is going to be palmitate, a C16 fully saturated fatty acid, and it may be elongated, it may have points of unsaturation added to it, it just depends on what the cell needs. But the liver says, I'm not going to just hold on to this free fat, I'm going to take that fat and I'm going to put it onto glycerol 3-phosphate, 1 fat, 2 fats, and now I've made phosphatidic acid. And then it can knock the phosphate off and put a 3rd fat on there, and it's made the triglyceride. And that triglyceride, of course, is going to be a much more efficient storage for those free fats by having 3 together on the glycerol, but still the liver says, I don't want to store this triglyceride, I want to export it. So the liver will make a VLDL particle, very low density lipoprotein species here. So we'll see this coming up in the lipoprotein lecture a little bit more, but these lipoproteins are made both in the rough ER where the protein components are synthesized, they're sent to the Golgi where the phospholipids are added and the proteins are processed a little bit, and they're also filled up with the materials that they're going to carry. And when you look at the lipoprotein species, you see that there's a shell of phospholipid, primarily glycerophospholipids, but I can't rule out the fact that there would be sphingolipids there as well, but typically glycerophospholipids. And it's going to make a shell or a bag, and then you're going to fill that bag up with these very hydrophobic triglyceride molecules. Now, if you recall, the head group part of a glycerophospholipid is very polar and very water soluble, and the lipids, by facing outward here, have their most polar surface in contact with the water. But the hydrocarbon chains, those 2 lipids that are attached to the glycerol backbone, are very hydrophobic, and now they're going to point in. So this is not a bilayer where you have two layers of phospholipids, but rather you have a monolayer with a polar exterior and a nonpolar interior. So these triglyceride molecules, which are extremely hydrophobic, are very, very happy inside there as well. And we haven't really talked much about it, but at the same time, the liver is going to be synthesizing cholesterol, and the liver says, my job is not to store cholesterol. I want to export it out to the blood so cells can have it if they need it, and that cholesterol is going to be packed into the VLDLs as well. And so when you see a VLDL particle like this, you're going to see cholesterol, which is in there, and that's being produced by the liver as well. And there's a couple of forms of cholesterol that we'll talk about in an upcoming lecture. And then the last thing to tell you about is this protein called apoprotein B100, which is kind of like the ID card. If you're a VLDL and you're out in the blood, how is anybody going to know who you are? Well, you wear an ID tag, and for this, the apoprotein B100 is the ID tag that says, hey, I'm a VLDL. There is a closely related lipoprotein called the chylomicron. Chylomicrons are made by the intestinal epithelial cells, and the chylomicron has the same function. It picks up dietary fat that is uptaken by those intestinal epithelial cells. You make an exterior surface of lipids. The inside is hydrophobic. You put triglyceride, fat-soluble vitamins, anything hydrophobic, cholesterol that's in the diet. And the calling card or the ID card for the chylomicron is an ApoB48. Very similarly related protein, but shorter than the ApoB100. But when they get out into the blood, chylomicrons and VLDLs behave almost identically in terms of delivering fat where it needs to go. Okay, so when you have these VLDL particles produced and released by the liver, where do they go? They're just going to float around in the blood, and if there's a cell that needs fat, that cell is going to have a receptor that can grab onto and catch that VLDL, or it could catch a chylomicron the same. They kind of functionally behave the same. And let's look at that process just a little bit more here. If you have triglyceride in the blood, that triglyceride, because it's hydrophobic, can't be free in the blood, but rather that triglyceride is found primarily in chylomicrons, which are made by the gut, or VLDL particles, which are made by the liver. And that VLDL particle, or chylomicron, is going to bind to an enzyme called the lipoprotein lipase. The lipoprotein lipase will grab these particles, and it will start to process the triglycerides and make them into free fatty acids. Well, let's look at that a little bit here in this picture. Here's a cell. In this case, it's an adipose cell. And you know that adipose cell is great for storing fat. So if a liver has made a VLDL particle, and it's sent it out in the blood under a high glucose, under a high insulin signal, that's going to tell the adipose tissue that it's time to store fat. So this plus here says insulin causes this LPL enzyme to move out to the outer surface of the cell and where it's in contact with the blood capillaries, and that LPL now being on the outside can grab onto these VLDL particles or chylomicron particles, and it can hold them and then process the fat that's inside them. So this is really complicated here. What this is showing you is whatever this particle is, this particle contains triglyceride. It could be a chylomicron, or it could be a VLDL, because these particles are really very similar in the way they function and how they interact with the lipoprotein lipase, the LPL. Now notice that insulin will cause the LPL to be moved out to the outer surface of the cell here, where it can grab onto these VLDLs or chylomicrons. And notice that the insulin now makes the LPL, the lipoprotein lipase enzyme, far more active in binding and digesting the triglyceride and the particles themselves. So insulin is a really powerful signal that tells adipose tissue it's time to store fat. And now we get to one of the biggest problems we have with the American diet. The American diet is so overloaded with carbohydrates, especially simple carbohydrates, and that causes such an amazingly sharp insulin spike, and the insulin persists for a very long time because of the huge amount of carbohydrate in the diet. And because of that, our adipose cells are supercharged to become fat storage cells, and they just grow and grow and grow and grow because we have so much excess carbohydrate, and our body is trying its best to preserve and store that energy and not lose that energy. Okay, so when the LPL does its work, then there's going to be triglycerides that are glycerol backbones with 3 fatty acids on them. The lipoprotein lipase, a lipase is an enzyme which will cleave a fat off of a glycerol backbone. And those free fats now are going to be transported into the cell, the adipose tissue, as a way to store them. And once they get into this adipose cell, the cell says, you know, I could store free fats, but that's not very efficient. What I need is a way to make the storage a little bit more compact, a little bit more efficient. And so these free fats now will be activated with coenzyme A to make fatty acyl-CoA, and then they're going to be added to this glycerol phosphate like we saw before, and that's going to be 1, 2, and then a third fat gets stuck on to make a triglyceride. Triglycerides are, you take 1 glycerol and 3 fats, that's 4 particles, but you combine them, they become 1 particle. And in terms of osmolarity and stuff like that, it's a much more efficient storage. Sorry about that. So where does the glycerol 3-phosphate come from? Well, insulin is going to stimulate this cell to bring glucose in and to run glycolysis, and that glycolysis will give the adipose cell the energy it needs for its normal function, and in the process, you'll see a good healthy amount of dihydroxyacetone phosphate being produced, and you have an excess of it. So that dihydroxyacetone phosphate can be reduced to glycerol 3-phosphate. Glycerol 3-phosphate is that starting material that makes the glycerol backbone for triglyceride formation. Okay, now what's going on out here? So you have that triglyceride, it's bound to the lipoprotein lipase. How is that bound to lipoprotein lipase? We will see this in just a couple lectures coming up, but there is a protein on the exterior of this particle called ApoC2. ApoC2 is the molecule that literally binds to the lipoprotein lipase to trap the particle and to give this enzyme access to the triglycerides on the inside. And that's what insulin is doing. Insulin's making the LPL far more active and far more receptive to the ApoC2 protein. So they stick together very nicely. Now the lipoprotein lipase, if you remember the game Pac-Man with a little yellow ball that would chop, 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 chop. And that's what I think of when I think about lipoprotein lipase. It's an enzyme whose active site just gets in there and it just cuts all those fats off of the glycerol backbone. And it's almost like you have a balloon that's absolutely full. And as you chop up the triglyceride on the inside of the balloon, and as you move the fats out, that balloon gets smaller and smaller and smaller and smaller. You're removing the contents, you're removing the triglyceride, and you're removing the fatty acids. So it's getting smaller and smaller and smaller and smaller. And eventually, this triglyceride will become, the particle will become so small that the ApoC2 no longer really has a good exposure. It doesn't really bind very well to the lipoprotein lipase. That thing will come off, and when the particle comes off, if it's a chylomicron, it's called a chylomicron remnant. Most of the fat has been sucked out of it, it's just the leftover pieces. If you're a VLDL particle, the liver put a lot of triglyceride into you, but it also put a lot of cholesterol into you. And when the VLDL loses its triglyceride, it becomes primarily a cholesterol particle, and that is what a low-density lipoprotein is. So VLDLs become low-density lipoproteins when they lose the majority of their triglyceride. We will develop this with the blood lipoprotein lecture coming up. And the other thing to tell you about is when the lipoprotein lipase cuts the fats 1, 2, 3 off the glycerol, the fat is what is imported into the adipose tissue. The adipose tissue doesn't care about glycerol. It makes its own triglyceride from glycerol 3-phosphate. And so the glycerol that's left over from pulling those 3 fats off is just going to be ejected into the blood. It's going to circulate, and the liver will pick that glycerol up and phosphorylate it, and then it can use it for a variety of processes. Okay, so that's a lot to take in for an adipose cell, but let's add one level of dimension here. Not just adipose cells, but muscle cells also like fat, especially cardiac muscle, slow twitch muscle, and even fast twitch skeletal muscle likes these particles. They have lipoprotein lipase that responds to insulin, and lipoprotein lipase will bring those fats in. They're going to be used for energy production in these muscle. Now what's interesting is the muscle lipoprotein lipase has a much higher affinity for these particles than does the adipose cell lipoprotein lipase. Okay, what does that mean? All right, if you compare a adipose cell and its lipoprotein lipase to a muscle cell and its lipoprotein lipase, you'll find the muscle cell has a much, much higher affinity for these particles than does the adipose cell. Does that make any sense? What does affinity really mean? Affinity says how much do two things like each other? How likely are they to bind together when the concentration gets smaller and smaller and smaller and smaller? And when you have a very high affinity relationship, it means even if the concentration is really low, that molecule can still bind because the affinity is high. And if the affinity is low, it means even if you have a fairly high concentration of the molecules, they really don't bind because they just don't like each other very much. Well, adipose loves these lipoproteins. It loves to get the fat and it loves to take them in. So I'm not saying that adipose has a low affinity, but what I am saying is muscle has a much higher affinity. And so as the concentration of chylomicrons, as the concentration of VLDL particles gets lower and lower and lower in the blood, eventually the level will get so small that the adipose tissue can no longer bind those particles because it just doesn't have enough affinity. But muscle will still bind these particles even when their concentration is very low, because you always want to use fat for energy whenever necessary. You only want to store that fat when you have an excess of fat. And so by the adipose tissue having a lower affinity, it will bind lots of fat when the fat is very high in very high levels, and muscle will bind it when it's also at a very low level as well. Okay, here's the lipoprotein lipase. I really like this. So here's a blood capillary. Here are these chylomicrons, or the VLDLs, tumbling along in the blood. They're just having a good day, running along the blood, just enjoying the river. And they come across this cell that expresses this lipoprotein lipase. And you see this is an enzyme that will extend up and above the surface of the cell, and they have an active site. And the protein ApoC2 is this little green ball. That's the protein that is on the surface of the particle, the lipoprotein particle, either chylomicron or VLDL. And that ApoC2 has a specific binding interaction with the lipoprotein lipase. And this is the one where muscle has a very high affinity between ApoC2 and lipoprotein lipase. And adipose tissue has an okay affinity, it's just not quite as good as muscle. But when those two proteins come together, the ApoC2 will trap this particle on the lipoprotein lipase. You see a bunch of them sitting there. And just like Pac-Man, they stick their mouth up into this thing and chop, 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 chop. They start chopping these triglycerides and making individual fats come off. It kind of shows you grabbing them here and then carrying them and putting them in there. That's true, but it's also they're in contact with the surface of the cell, and you see free fats going across the membrane. And here's the glycerol. The glycerol actually gets ejected. You don't really need the glycerol, but you do need the fatty acids here, and the fatty acids are what are going to stay inside either the adipose tissue or inside the muscles tissue for oxidation and energy production here. Really, really nice picture. And you see that these things are full of triglyceride. The particle is very big. And as the lipoprotein lipase degrades and consumes these triglycerides, then the volume inside that bag will get smaller and smaller and smaller, and the particle will get smaller and smaller and smaller. And either the chylomicron will leave as a remnant or the VLDL will leave as an LDL particle. So up to this point, we've been talking about insulin and how insulin is a signal to store and to synthesize and to build up reserves. But now let's talk about the glucagon and fasting signal. If you recall, adipose is going to store fat, and it stores fat primarily for the purpose of storing it until it's needed during fasting. And of course, during fasting, glucagon is going to be the hormone in control. During fasting, we have a high level of glucagon. We have a low level of insulin here. So high glucagon is going to stimulate the glucagon receptor, G protein, adenylate cyclase, cyclic AMP, protein kinase A, like we've seen before. And one of the targets of protein kinase A is an enzyme called hormone-sensitive lipase, or HSL, hormone-sensitive lipase. So when the lipase is dephosphorylated, it's inactive. It's just sitting there doing nothing. But when glucagon signaling starts, this protein gets phosphorylated, it becomes activated, and the hormone-sensitive lipase will take triglycerides, and it will break those 3 fats off and leave a plain glycerol with the 3 fats, and they are all exported out of the cell. Now we know that fatty acids are hydrophobic and they don't do well in an aqueous medium, and so the protein albumin will be sitting there on the surface of the cell to pick up these fats and carry them safely in the blood to the tissues where they need to be. Glycerol, of course, is a very polar molecule, and it's soluble in water, and it's just going to get into the blood and circulate and be picked up by the liver. When you're doing fasting, it's going to be picked up by the liver for gluconeogenesis. So the main thing here is glucagon gives a fat liberation pathway here, and that fat liberation is the hormone-sensitive lipase degrades triglycerides to free fats and glycerol, which are exported here. Now, there's a cool thing about adipose tissue that I really like, and that is adipose tissue is smart. It says, I have got a lot of fat stored here, and I can give you a lot of fat for a lot of days. I can really hold out for the long haul here. And the adipose tissue needs to make a decision. How much fat do I release, and how fast do I release that fat? And that's what this slide is showing you right here. We know that under a glucagon signal, the hormone-sensitive lipase right there, the hormone-sensitive lipase under a glucagon signal is activated. It's going to break down the triglyceride to free fatty acids. And of those free fatty acids, it doesn't just let them all out automatically, but rather, about half, 60% or so, it's okay, are going to be released. And of course, the corresponding amount of glycerol is going to be released as well. So that means that the adipose tissue keeps a couple of free fats, or a small percentage of the free fats, in reserve. And if glucagon signaling is persisting, it's going to release more fat. But if the glucagon signal starts to decrease, if insulin starts to come up, and the glucagon starts to go down, then it can retain those fats. It's not going to release those excess fats in the blood just for them to be wasted when insulin comes back, but rather by holding them in reserve here, it's much easier to cycle those free fats back to synthesis of triglyceride if an insulin signal comes. So that'd be, for an example, you've had breakfast, and you maybe had some carbohydrates, and so you've got insulin in your blood, and blood glucose is a little bit high. That's fine, no problem. But after 4 hours, when you're done absorbing and you're done processing all that, and everything is absorbed by the cells, the insulin levels down, the glucagon levels back up, and you're in the fasting mode. Okay, that's fine. Hormone-sensitive lipase is doing its thing. It's liberating free fats. But within just a short period of time, it's time for lunch. And you have maybe a little bit of carbohydrates, maybe a little protein, a little fat, and that carbohydrate is enough to stimulate insulin again. Well, boy, it sure is lucky you didn't release all that fat because you didn't need to, because more food stuff is coming and more insulin is coming. So luckily we didn't lose that fat. And because insulin is present again, you can synthesize. And just like we said before, where's the glycerol 3-phosphate coming from? Well, under a glucagon signal, when you have free fatty acids being produced by hormone-sensitive lipase, glucagon doesn't favor glycolysis, but rather glucagon will favor gluconeogenesis. And in fact, adipose tissue will do half of gluconeogenesis in order to produce the DHAP necessary to serve as starting material to make glycerol 3-phosphate here. So as we've seen before, any one of the 3 precursors come in through the first bypass here. You go through that first bypass, there's that PepCK, you make phosphorenylpyruvate, and that's going to go backwards all the way up here to glyceraldehyde 3-phosphate, DHAP, and that DHAP is what is grabbed to make the glycerol 3-phosphate. So you're not really doing all of gluconeogenesis. You're not trying to form glucose, but rather you're trying to form an intermediate glycolysis that you can use to make that glycerol backbone. Okay, so jumping back now to glycerophospholipids and sphingolipids, if you can build them, you need to degrade them. And as you can imagine, we have membrane components in our cells that have the bilayer and all those lipid species. And sometimes it's necessary just to recycle. You want to pull those lipids out and you want to degrade them and you want to put new lipids in their place and refresh the membrane. If you do that, you need to send the glycerophospholipids and the sphingolipids to the lysosomes. And the lysosomes are going to contain all of the enzymes necessary to degrade these cellular components. And we've seen these before. We saw this in the lipid lecture, I believe. But there are 4 enzymes that serve to break down this glycerophospholipid. There's a phospholipase A1 and A2 that are going to cut the ester bond between the fatty acid and the glycerol backbone. That'll liberate free fats, and those free fats then get released into the cell. And then you have phospholipase C or phospholipase D. Phospholipase C cuts between the head group and the phosphate, and that gives you the head group with the phosphate on it. Or if you have phospholipase D, that's going to cut between the phosphate and the head group, and that's going to give you a phosphorylated glycerol and the head group. Both of those molecules can just be released by the lysosome into the cell for recycling here. And there are many diseases that are caused by defects in lysosomal enzymes, especially ones that break down sphingolipids and glycerophospholipids. We talked about glycogen degradation issues in the lysosomes. You see the lysosomes are really, really important for degradation. If those enzymes aren't present, then it causes a lot of trouble for the cell. And in terms of the glycolipids, there's a whole bunch of enzymes that are present in the lysosomes to break down all of these really strange lipid molecules that we have. And you see there are specific diseases that correlate with specific enzymes that are deficient that changes the degradation of these lipid species. So I really don't want you to memorize this. You don't have to learn this table at all. I just want you to be aware of how incredibly easy it is to have a disorder because you simply don't have an enzyme that's needed to degrade a certain molecule in the lysosome. All right, let's do a sample question. Glycerol can serve as a starting point for triacylglycerol synthesis in liver, but not in adipose tissue. What is the starting point for triacylglycerol synthesis in adipose tissue? Wow, well, you know, liver is lucky because liver has that enzyme glycerol kinase, and that's the easiest thing in the world. It's just to pop a phosphate onto glycerol and make glycerol 3-phosphate. But adipose tissue does not have glycerol kinase, so what's it going to do? It needs to start with some molecule that is produced naturally that's kind of close to glycerol 3-phosphate. And the question is, do you remember what that is? Well, if you think about it, I think it was DHAP was that molecule that's produced in large measure either by glycolysis, making DHAP that can be used to synthesize triglycerides in adipose tissue, or during fasting, you produce DHAP by gluconeogenesis, which will be the glycerol backbone, which is used to make triglycerides. So we're looking for dihydroxyacetone phosphate or something like it, and there it is. An obese teenager presents with a metabolic disorder manifesting as an inability to liberate free fatty acids from triacylglycerol stores in adipose tissue. Well, that's a big sentence. So a teenager that's overweight, there's obviously a metabolic disorder, and the adipose tissue can't release fats, and that's part of the reason why the fat cells are so big, and it's adding to the obese nature of the teenager. Okay, so what's going on? Clinical tests show that the patient is able to digest, absorb, utilize, and store dietary triacylglycerols and fatty acids normally. Wow, okay. So that means their LPL enzyme is okay, their ApoC2 is okay, chylomicrons are okay, VLDLs are okay. You get those fats going into the adipose tissue. The adipose tissue has glycerol 3-phosphate. It can build the triglyceride. It can store the fat. So obviously there's nothing wrong with putting fat into the adipose tissue. It sounds to me like the problem is breaking down those triglycerides and getting the fat out. So, however, following a fast and a glucagon challenge, and that's sort of clinical speak for a person who's fasting to make sure they have a basal level of glucose, 80 to 100 milligrams per deciliter, and that's going to allow glucagon to be the hormone in charge. So that's the glucagon challenge. And so during that glucagon challenge, you don't see free fatty acids coming into the blood system. So whatever it is preventing the adipose tissue from liberating these free fatty acids and releasing them into the blood. Which enzyme do you think is the problem? So there's two ways to do this. One of them is just to simply go through each answer choice and say, could this be it? Why or why not? And that's certainly a good way to do it. The other way to do this is to say, am I aware of one enzyme in adipose tissue whose job it is to break down triglycerides and make free fats? And if you can remember the name of that particular enzyme, then all you have to do is look at the answer choices and see if you find it. So we can do both. Let's start with the latter one because it's a little bit better. And that is, I know that hormone-sensitive lipase is the enzyme that responds to glucagon, that's the hormone, telling it to be active, and it breaks down triglycerides and makes free fats. And those free fats are released into the blood. They're carried by albumin, but they're still free fats in the blood. Okay, so obviously this person has trouble with their hormone-sensitive lipase. So when I go and I look at the answer choices here, then I see hormone-sensitive lipase, and I know I can pick that one right away. But if you don't remember that answer choice, then you can go through the answers together. You can say acetyl-CoA carboxylase. Well, that's an enzyme that's involved in fatty acid synthesis. So all the synthesis stuff up here was perfectly normal, but what's wrong is getting the fats out of the adipose. So clearly, answer A is not a choice. Lipoprotein lipase. Well, we know that that's normal because, again, the stem up here says that everything is just fine as far as getting fats into the adipose tissue, and that's what lipoprotein lipase does. It gets fats into the adipose for storage. It has nothing to do with bringing fats out during times of need. And we haven't talked about lecithin cholesterol acetyltransferase, but we will very shortly. But that's a way we stick a fatty acid onto a cholesterol molecule to make it more hydrophobic, and that helps its transport properties. The exam for this comes after the cholesterol lecture, so you'll get this enzyme in that lecture coming up. So really, hormone-sensitive lipase becomes the only really sensible answer that there is, and in fact, that is going to be the correct one. Okay, here's a patient that's very concerned that his adipose cells cannot import glycerol from the blood, since he knows glycerol is the backbone of triglycerides. What can you tell him that will allay his concerns? He's so worried about his ability for adipose to store the fat properly, and he's worried about that. And so you think, well, is glycerol really what the cell imports? Do you need glycerol to come in so you can attach fats to it? And you remember, no, that's not the case at all. Adipose tissue is going to use DHAP, which is produced by glycolysis during the insulin signal, or it's produced by gluconeogenesis under the glucagon signal, and that is going to be the backbone for making triglycerides. So you're looking for an answer that has something to do with DHAP being used for triglyceride synthesis. And very quickly my eye jumps to answer number C, because his adipose cells can use DHAP as a starting material for the synthesis of triglycerides here. A patient has a PEP-CK, phosphoenolpyruvate carboxykinase defect in his adipose cells. What will be the effect of this defect on adipose cell function? And you're thinking, oh my gosh, phosphoenolpyruvate carboxykinase is a gluconeogenesis enzyme, and the liver does that to produce glucose to keep blood glucose levels where they need to be over longer fasts. Okay, that's true, but adipose tissues also do gluconeogenesis because they need to produce a certain amount of DHAP that can be used to recycle the excess free fatty acids that were produced by hormone-sensitive lipase, but were not released into the blood, and you want to turn those things back into triglycerides. So you're looking for an answer here, something that has something to do with triglyceride formation. So glycerol in his adipose cells will not get phosphorylated to support triglyceride synthesis. Well, in the adipose cells, glycerol does not get phosphorylated. Adipose cells do not have glycerol kinase, so that's not even a viable answer. His adipose cells will not bind VLDL or chylomicrons properly. Well, that's going to be due to the LPL, the lipoprotein lipase, and that apoprotein C2. So again, that has nothing to do with gluconeogenesis or triglyceride formation. His adipose cells will have a decreased ability to synthesize DHAP needed for triglyceride formation. Now that sounds pretty interesting, because if you're synthesizing DHAP, the way to do that is gluconeogenesis, and DHAP is that sort of central intermediate there, and PEP-CK is going to be that first bypass enzyme of gluconeogenesis. So under these conditions, if there's a PEP-CK defect, the cell can't use lactate, the cell can't use alanine as a starting material, and it can't use glycerol either because it can't phosphorylate that glycerol. So you see adipose tissues would have quite a big deal of trouble here if there's a PEP-CK dysfunction. His adipose cells will not be able to carry out gluconeogenesis. Well, that's not really the idea. The idea is to carry out the first half of gluconeogenesis, not all of gluconeogenesis ending in glucose. And his adipose will not export free glycerol. And again, that's not really the issue here either. You're not trying to produce glycerol, you're trying to produce glycerol 3-phosphate that is used to make a triglyceride so that you can reincorporate those free fatty acids for storage. How about one more? Here's a patient with a disorder in her adipose lipoprotein lipase enzyme, where the enzyme does not recognize ApoC2 on the surface of these VLDL particles or chylomicrons. Which of the following would be a likely consequence of this disorder? So you have to read all of the answer choices to figure out which of the consequences would be correct. But the first thing you want to do is you say, what's the function of ApoC2 on the surface, and what would happen if that ApoC2 doesn't work properly? And you remember, ApoC2 is that protein on the outside of the lipoprotein particle that will bind to the LPL enzyme and trap that particle so that the LPL active site can stick its mouth into that particle and start digesting those triglycerides. And so if the LPL enzyme doesn't recognize ApoC2, it can't bind ApoC2, so the lipoprotein particles will not stick to the lipoprotein lipase enzyme, and it can't degrade them, it can't get the fat in. So you're going to be looking for some kind of answer that has something to do with the fact that they can't bring in the fat into the cell the way it's supposed to. So let's look at these different answers. Her adipose cells will be much smaller than normal. Does that make sense? Well, it sure does. Lipoprotein lipase will bind the VLDL, and it will bring the fat in, and then the fat is put into triglycerides, and that's going to cause the adipose tissue to swell and get bigger because it's incorporating more and more and more fat. And if you can't bring the fat in from the VLDLs, then the cells won't grow. They won't incorporate fat. So that sounds pretty good. Her blood lipoprotein level will be much higher than normal. Whoa, that sounds like a good answer too. When the ApoC2 sticks to the lipoprotein lipase will degrade those particles and turn them into remnants, and those remnants will be recycled. And if you don't do that, then nothing ever happens to those particles, and they stay in the blood, so the concentration is higher. Her blood vessels will have increased level of fatty streaks. Well, we haven't talked about that yet, but when we talk about cholesterol and LDL, we'll see what a fatty streak is. And we'll find out that if there's too much lipoprotein in the blood and they don't get consumed by the cells, they have a horrible habit of aggregating, sticking together, sticking to the wall of the blood vessel, and making what's called fatty streaks, which is the beginning of vascular pathology. Her liver may have more fatty acid deposits than normal. Huh. Well, that sounds right. If your liver is producing VLDLs and exporting it, and it gets in the blood, but the VLDL never leaves the blood, then the concentration of VLDL will be much higher in the blood, and the liver will recognize that. It will say, wait a minute, there's already a lot of VLDL out in the blood, and I'm making more VLDLs, and I'm trying to push them out in the blood, and it's hard for me to send VLDLs out when there's already so many out there. And so the liver says, well, I can't move this VLDL out, and since I can't move it out, it's going to stay inside the liver, and they're going to start to aggregate. They're going to start to form these little fatty patches inside the liver, and that leads to what's called a fatty liver syndrome. And that is exactly correct. So E is all the listed answers are likely consequences, and that's the case. All of these things can happen when you have a simple disorder where your lipoprotein lipase can't bind and digest these particles. Okay, that should do it for this one. So I say thank you, and we'll see you on the next one.